Okay, so are you guys all ready?
Ready?
All right,
Let's go.
good. We'll open up the waiting room and get started. Let's see. Okay, so welcome and thank you all for joining us today for another session of the PCE3 seminar series. And if you don't already know, PCE3 is a NASA RCN that stands for Prebiotic Chemistry and Early Earth Environments. And one of the main goals of PCE3 is to bring together researchers from the various disciplines who work together towards better understanding how life may evolve on Earth. And so to that end, we're kind of interested in whether this seminar is actually reaching researchers from different fields. So we have a short poll that we're going to put up so we can get kind of an idea of all your interests. So let me bring that up really quick. So that poll should have popped up. And while you're taking a look at that, I'll mention that another goal of this seminar series is to highlight some of the exciting work being done by early career researchers. So today we're going to be hearing from Harpreet Kaur of the University of Strasbourg and Ziwei Liu, who works at the MRC LMB at the University of Cambridge. And so one quick note before we get started is that we're going to keep the chat closed to questions until the end of all the talks. And so try to keep a note of any questions that pop up during any of the presentations. And also, before we get started, I'm going to end this poll right now. So let me just close that. So before we hear from Harpreet and Ziwei, we'll have a short introduction of today's theme, which is proto-metabolism, given by Kamila Muchowksa. And she's a CNRS Research Assistant Professor at the University of Strasbourg. And she received her PhD from the University of Edinburgh. And after this, she spent a few years as a postdoctoral researcher studying metabolism-like reactions potentially related to the origin of life. So thank you for joining us today, Kamila, and go ahead.
Thank you for this very kind introduction, and I'm really grateful for this invitation. Really excited to be here. We are up for something really, really nice from our two speakers later on. And before we start that, let me just get started with some very, very basic definitions. So I'm going to share my screen and hope you will be able to see the correct one. Okay, you can see my PowerPoint, I hope.
Yeah.
Now, please nod if this is the right view. Are we good?
Everything's
okay.
Good. Looks good.
Everything good, perfect. So everyone, the theme of today's seminar is proto-metabolism. So before we jump into proto-metabolism, let's just set the scene first. So the two speakers in today's session work in the area of prebiotic chemistry, and the goal of prebiotic chemistry as a science is very hefty, and it is as hefty as it is fascinating. Because in order to truly understand life, we need to be able to explain how life came into existence. So in other words, the question prebiotic chemists are trying to answer is how geochemistry of the early planet Earth, some 4.5 billion years ago, became biochemistry, which you have in this circle. And biochemistry is essentially what governs life. So this is how we currently depict biochemistry. It's an incredibly complicated map of chemical transformations. We see lots of patterns, circles, lines. And this map describes everything that keeps life alive. It is a map of the entire metabolism that happens every second in a cell. So the metabolic map is indeed very complex, and to be able to talk about proto-metabolism later on, we first need to become acquainted with some fundamental concepts and definitions. So what do we have here? Metabolism in biology is defined as a collection of chemical processes. These processes form building blocks of a living system from often very simple materials. So these materials are a food set for an organism. And these processes connect the internal maintenance of the living system to an external energy source. So this is a very, very broad and general definition of what metabolism in life is. So our biochemical metabolism can be divided into two different types of reactions. So on one hand, we have anabolic reactions, and these are those that build up macromolecules from small building blocks, such as fatty acids, amino acids, sugars, and this is usually helped by chemical energy delivered by ATP or cofactors in biology. And as these reactions need energy, we call them endergonic. On the other hand, the formation of molecules that provide the chemical energy, this proceeds through exergonic catabolic reactions. So here we have high energy molecules that are breaking down to small molecules like carbon dioxide or water. So today, all these reactions form a complicated metabolic network. And in biology, a huge majority of these reactions is promoted by enzymes. And these enzymes are really big. They are proteinic enzymes, like the one that you see on the right-hand side of the slide, rotating a beautiful blue ribbon. These enzymes oftentimes use ATP as energy source. So of course, these large molecules did not exist on the early Earth billions of years ago. And this is one of the few points about which prebiotic chemists agree quite unanimously. So another important point is that the prebiotic version of this metabolism, so before life existed and before these enzymes had evolved, this is what we call proto-metabolism, the predecessor. So especially in its early stage, the proto-metabolism must have been much simpler than the one that we observe today. So clearly there was a point in the process of life's emergence when prebiotic chemistry reactions were promoted by catalysts, and these catalysts were available in their environment. For example, very geochemicals, and these were clearly not these complex proteinic enzymes. The energy for these reactions of proto-metabolism was not sourced by ATP, or at least not immediately, but what different prebiotic chemists propose ranges from the UV light to pH or redox gradients to spark discharge to radioactivity even. So certainly, as always, when we can't really go back in time and check with our own eyes what really happened, whenever prebiotic chemists think of the chemistry that makes up life, they always run into this chicken or egg problem. And what it's about, the catalysts that help build metabolites, today they are enzymes, which themselves are proteins, which are built from amino acids, aka metabolites. And it gets even worse because the information to make those enzymes, this information lies, it's coded by the genetic polymers, which polymers themselves are built from transformed amino acids and sugars, and these are also metabolites. So it gets really quite convoluted and quite complicated. But as we are scientists, the correct answer to the chicken or egg problem is a dinosaur. So in the broadly understood origin of life field, approaches to proto-metabolism vary a lot. And one particular point that I would like to bring to your attention is the level of parsimony or similarity between the metabolism today, between biology, and the proposed experimental reconstructions of proto-metabolism. I have quoted two prominent scientists in the field who have quite different views at first glance. So the first quote is from Nick Lane: "To understand the origin of life, it would be foolish to ignore life as a guide." And on the other hand, we have a quote from Ram Krishnamurti, that there may be no way to proceed all the way from biology to prebiotic chemistry using biological processes as the guide. So you could think that these seemingly opposing views are something really bad to have in the field, if they're going to argue. But actually, firstly, they don't always have to be mutually exclusive. And secondly, challenging each other's hypotheses and discussing our arguments and rethinking our options while pursuing the same goal at the same time can only lead to better science. So with this, I will give up the stage to today's speakers, Harpreet and Ziwei, who are going to present the takes on this mysterious yet truly remarkable process, the emergence of proto-metabolism. Thank you.
Great. Thank you, Kamila. That was a great kind of introduction and setup for our upcoming speakers. And so next we'll be hearing from Harpreet Kaur, who is currently a PhD student in the prebiotic chemistry at the Institute of Supramolecular Science and Engineering in Strasbourg. And she's working with Professor Joseph Moran. And Harpreet's research has focused on prebiotic chemical processes that could have contributed to initiating self-organized prebiotic chemistry on early Earth. And so thank you for joining us, Harpreet, and go ahead.
Okay, thank you. So I'm gonna share my screen now. Okay. Do you hear me well?
Yeah, everything looks good and your mic sounds good.
Okay, perfect. So thank you, Kamila, for the introduction, and thanks to the organizers for inviting me to speak about my research on how hydrogen gas and transition metals could have powered protometabolism on the early Earth. As the subject of today's session is protometabolism, so I would like to talk about what protometabolism is. But before engaging into this specific concept, I would like you to ask this general question: what is life? Over hundreds of years, scientists have tried to find an answer to this question, and we can say that life, from the simplest bacteria to human, is based on a very complex network of chemical transformations. Everything related to life, or everything characteristic of living organisms, such as breathing, reproduction, etc., are all the final effect of a fine-tuned set of chemical reactions. And a sum of all is called metabolism. So it's all the processes that build up molecules, break them down, and everything in between. And such kind of chemistry is relying on catalysis, acid-base interactions, redox reactions, and so on. So we can say that life is a self-organized dynamic chemistry. Protometabolism then is its precursor. It was probably much simpler, but no doubt still highly complex. Before I go further, it is worth remembering that organic substances which make up living organisms are not different from inorganic ones, and they follow the same laws. Now, this brings me to the question that the Moran lab wants to answer: how did such a complex, self-organized chemistry get started spontaneously on the early Earth, where we had only inorganic and simple molecules available? We will dig into this question during this session, but first, I guess it is helpful to look at other much simpler systems that exhibit spontaneous order. Here you see an example of hurricane. So what happens is air and water self-organize into a hurricane only under very specific conditions. A specific energy source, namely a temperature gradient, is needed, but only a specific gradient will make it happen, and only a specific distance scale. In the similar way, we can argue that synthetic chemistry should also self-organize to form a proto-metabolism under very specific conditions. As I said earlier, metabolism is driven by reducing agents, oxidizing agents, enzyme catalysts. Likewise, to allow proto-metabolism to form spontaneously, we should expect that specific reducing agents, oxidizing agents, specific inorganic catalyst would have been needed. We don't know which reducing agent drove protometabolism, but we know quite a bit about which reducing agent probably drove the metabolism of early organisms, and it was none other than the hydrogen gas. This is what we know from the phylogenetic and thermodynamic studies. Phylogenetic studies, they tell us about the enzymes, which were found to be present in most ancient organisms, and these studies show that these enzymes largely promoted the reactions using hydrogen as a reducing agent and carbon dioxide as a building block. Thermodynamic analysis of the most ancient metabolic networks also point at hydrogen gas as the most likely driver. Of course, we cannot be sure that hydrogen drove protometabolism, but based on what we know so far, it seems like the most parsimonious choice. It has many characteristics which makes it a kind of perfect candidate for driving a system like protometabolism. For example, it's a gas, so it can be easily transported. It's an extremely simple reducing agent and produced by numerous geological processes like serpentinization and water radiolysis. And it's also chemically inert in the absence of specific catalyst. Okay, now we know what kind of conditions we should look at. But before we set out to search for those conditions, we have to decide what dynamic chemistry we are looking to find, as the possibilities are nearly endless. So to simplify our search, we have made the assumption that there was a continuous, unbroken progression from the first self-organized chemistry to the metabolism of ancient organisms. Just like in the same way as there has been a continuous progression from ancient organisms to the organisms alive today. Now, this is an important assumption to consider. We don't expect protometabolism to be identical to the chemistry of core metabolism, but at the same time, we cannot expect it to be radically different either. I hope you are getting my point. What I'm trying to say here is complex synthetic networks that support all of life's functions can't take a break. So this constrains how much they can change. For that reason, in protometabolism, we can still expect to find most of the same simple metabolites that we find in ancient metabolism and the same classes of reactions that form them. Here I show you a rough roadmap for what we are looking for. So we would expect some reactions that reduce CO2 into metabolites such as ketoacids, similarly to the most conserved and ancient CO2 fixation pathways, which is acetyl-CoA pathway and RTCA cycle. Then we would expect some chemistry that interconverts the formed ketoacids into amino acids, and then those amino acids to react further to make other complex molecules such as ribonucleotides. So this is the basic concept we should look at. Let's have a look what have been done so far. So over the past 5 years, our group and other groups, such as the group of Professor Martin, Tutsius, Ralser, Krishnamurti, Barge, Lane, and others, have reported conditions that make many of these pathways occur in whole or in part without enzymes. I must say these results offer a preliminary indication that such kind of approach can work, but the problem remains far from being solved. Now again, just to fresh our minds, what's the problem? The problem is to get a self-organized autonomous chemical network which can mimic biological pathways. So we need a self-organized biology-like system, and the conditions that were found here are often fairly different and have different driving forces. As you can see, some are driven by metallic iron, others by organic reducing agents, or even by radical chemistry. So all these different chemistries cannot give rise to autonomous system that we are looking for, that we are talking about. To have a self-organized protometabolism, we would want to see a single reducing agent, ideally hydrogen, that can drive multiple processes under the same conditions at the same time. We would want to see milder conditions compatible with all the biomolecules necessary to life. This way, all these processes could work together and arrange as a network. But this is not what experiments have found so far. Maybe the problem isn't the experiments themselves, but us, the experimenters. Perhaps we limited our mindset about what we consider as prebiotic reagents. So the next question arises, what or which are the limitations? These limitations are our assumptions. One of the first assumptions that we usually make is regarding the nature of the inorganic catalyst that we screen in our experiments. Many of the enzymes that catalyze the reactions of core metabolism use geologically abundant transition metals in their active sites. So intuitively, in a bottom-up approach, we normally think that the mineral forms of these same metals were the original catalyst for the same reactions in protometabolism. So this hypothesis, this assumption centered around earth-abundant metals like iron and nickel. Now, I'm not saying that this assumption is wrong, but unfortunately, this thinking hasn't yet yielded a sufficiently complex metabolism-like networks. There is something missing. We need to find that missing piece, which along with other pieces can bring us more closer to the bigger picture of protometabolism puzzle. Now you can say, okay, we need to broaden our mindset, but what we've missed and where to find that missing piece? To answer this, let's have a look together at the history of evolution of catalysis by humans to see if we can learn anything or see if we can find the missing piece. Here you can see an alkene hydrogenation catalyst as an example. Interestingly, the first catalysts described here for this reaction were rare metals like palladium and rhodium. No special ligand was required, they just worked in their native metal forms. It took nearly 80 years before humans developed well-defined hydrogenation catalysts based on abundant metals, whose efficiency is on their highly complex organic ligand. What we learn here is we started with the metals that are inherently great catalysts, even if they are rare. Then, as the supply of these precious metals was low, with time, we started to replace them with catalysts based on more abundant metals. But where more organic complexity was required to have the same functionality as the rare ones. Again, the question to be raised is, if prebiotic chemistry self-organized around inorganic catalyst, why wouldn't it have followed the same logical progression? What if the very first ancestors to the complex metalloenzymes were not based on abundant metals at all, but simply on rare metals? It's possible that on the early Earth, once the reaction networks catalyzed by rare metals produced complex organics, those organics then in turn functioned as ligands to turn abundant metals into active catalysts, resulting in the abundant metal-based metalloenzymes that we see today. Okay, great. You might say, this is a crazy idea because metals like palladium or rhodium have a natural abundance on the ppb range, so it doesn't make sense to even consider them. But at the same time, I would point out that self-organized chemistry doesn't need to initiate on the global scale. It is well known that precious metals accumulate in much higher concentrations locally. So even if we have lower global concentrations, there are places where the local concentration is high, and the same would have been true on the early Earth. Here's an example to support this idea. Samples of mineral called pentalendite have been identified in Russia, and this contains up to 25% palladium by weight, which is a lot considering the fact that rare metals doesn't need to be used in stoichiometric quantities to exhibit their catalytic activity. Just traces are enough. And since we are talking about self-organized chemistry based on abundant gases like hydrogen, carbon dioxide, or carbon monoxide, the chemistry could take place wherever the best catalysts are to be found. So with this idea and all the necessary information in mind, we decided to investigate a number of systems for how ppm level of precious metals might influence the chemistry. I'm going to walk you through all these 4 stages of complexity step by step. So please stay with me. We will go from CO2 fixation to keto acid synthesis, from keto acids towards amino acid synthesis, and from amino acids towards precursors to ribonucleotide synthesis. Now, the first step to investigate is CO2 fixation, as this is the first step, or better I call it as first bottleneck of protometabolism, and it's widely thought to be the most ancient CO2 fixation pathway in biochemistry. We've previously shown, together with the groups of Professor Martin and Dr. Tucci's group, that CO2 and H2 react over various iron and nickel-based catalysts at high temperatures to give CO2 fixation products. But as I said earlier in the introduction, these conditions are not compatible to allow further complex chemistries. Therefore, we needed an alternative. We decided to investigate this reaction between hydrogen and carbon dioxide under extremely mild conditions using rare metal catalysis. This work was done by my colleague, Emily Werner. She used room temperature in pH 8 water under 5 bar of hydrogen gas, bicarbonate as CO2 source, and just 32 ppm of commercially available palladium catalyst. These are extremely mild conditions, and yet the reaction is extremely efficient. After 24 hours, it produces 150 millimolar of formate. That's a stunning 52% yield of all bicarbonate solution that is reduced to formate. And it means that there is 520 times more product formed than catalyst put into the reaction. Other important factor of this reaction is it's extremely clean, therefore can be easily integrated to other chemistries. Also, the reaction is counter-ion dependent. It produces more formate in the presence of ammonium as counterion, indicating importance and different efficiencies of various inorganic salts in prebiotic context. Then we study the second system, the reaction network between oxaloacetate and glyoxylate, which could bring us to the all-universal core metabolic keto acids formed in RTCA cycle and which are crucial for the synthesis of other biomolecules. Overall, this subsystem functions as the one carbon homologue of the reverse Krebs cycle, all composed of keto acids. Let's have a look on how does this network work. Oxaloacetate and glyoxylate undergo a decarboxylative aldol reaction to give malloyl formate. Then this is followed by a dehydration to give fumaroyl formate, an alkene reduction to give alpha ketoglutarate, and again a second aldol reaction to give isocitroyl formate. And then a second dehydration to give aconitroyl formate. Pyruvate and citroyl formate can also form from the decarboxylation and decarboxylative dimerization of oxaloacetate respectively. This network was previously reported by our group and also by Krishnamurthy group. Both our experiments and those of Krishnamurthy lab had been done at high temperatures and at high catalyst loadings. We used iron and other group used phosphate buffer to induce acid-base catalysis. The reason these harsh conditions were required is because the slowest step in the reaction sequence is the reduction of the alkene in fumaroyl formate to ketoglutarate. So barrier to the reduction is high, therefore high temperatures are required, but these are not the conditions that we want. If we perform this network in the presence of ppm levels of palladium, bicarb hydrogen or formate as the reducing agents, interestingly, the entire reaction sequence can be observed even at room temperature at neutral pH without any added buffer. So there is a catalytic palladium hydride species that is formed and is able to reduce the alkene under these conditions. Now that the reaction occurs at room temperature, there is very little decomposition, and the same reaction carried out in the absence of palladium catalyst still produces the initial products of the aldol condensation, but none of the products downstream of fumaroyl formate, which suggests the role of palladium in the reduction step. Then with these conditions in hand, we wanted to study the next subsystem, which is amino acid synthesis. Before I tell you about our our experiments, I just want to give you a little background about how amino acids are made in biology from the corresponding keto acids formed in the reaction network that I just showed you before. So typically, ketoglutarate undergoes a reductive amination reaction using ammonia and a biological reducing agent to give glutamic acid. Then glutamic acid undergoes transamination reaction to transfer that ammonia to other keto acids, converting them to amino acids. The mechanism and inputs of these two processes is different, but the overall result is the same. In the end, the keto acids are converted into amino acids by a formal reductive amination. Some work has been done on the prebiotic reductive amination using iron minerals as reducing agents. But the problem is, mostly in all the cases, a highly concentrated ammonia solution was required as well as high temperatures. Unfortunately, oxaloacetate does not reduce aspartate under these conditions because it decarboxylates much faster than the reaction occurs. And we need aspartate synthesis since aspartate is the biosynthetic precursor of ribonucleotide biosynthesis. Taking this into account as a different approach, our group showed that bifunctional amines such as hydroxylamine and metallic iron can allow the reaction to occur on all of these keto acids. But still, it's not a good solution, as hydroxylamine is not very geologically realistic, and because it is such a good nucleophile that it reacts with all activated acyl species, such as acyl phosphate or thioesters. And we don't want just a soup of reactions, but a self-organized chemistry. And this brings us to the experiments done on our third subsystem. Again, we used palladium as catalyst, 32 ppm in the reaction mixture, hydrogen bicarb or format as a reducing agent, and everything was kept at neutral pH for 72 hours. We found the reaction to be compatible with different keto acids using only 5 equivalents of ammonia. We obtained all 4 core metabolic amino acids, glycine, aspartate, alanine, and glutamate, along with other biologically relevant amino acids such as valine, leucine, and isoleucine from moderate to high yields. Important thing to notice here is reductive amination occurred selectively in comparison to corresponding hydroxy acids, which were just found in very small quantities in the reaction mixture. In brackets, you can see the ratios between amino acid and hydroxy acid formed in the reaction. I would also highlight that this set of conditions, unlike the chemistry developed so far, are compatible with oxaloacetate to give aspartate, which is an essential biosynthetic precursor to RNA. Now, having amino acids in hand, we are progressing towards our fourth system, towards ribonucleotides. Till now, I've showed you that we can reduce CO2, drive a 1 carbon homologue of the reverse Krebs cycle, and perform reductive amination of keto acids, all at room temperature near neutral pH using trace amounts of palladium as a catalyst. Then we were interested to find out whether the N-carbamylation of amino acids might occur at room temperature and if it's compatible with the conditions reported in other subsystems. I would underline again that N-carbamylation is an important step in pyrimidine ribonucleotide biosynthesis. Previously, our group had reported this reaction at 60 degrees, but we didn't know if it could occur at room temperature. And we've found this happen to be possible. For example, exposing glycine, aspartate, alanine, and glutamate to carbamyl phosphate at room temperature in neutral pH water cleanly gave the corresponding N-carbamyl amino acids, producing up to 50% of yields, with only unreacted amino acid remaining. The production of carbamyl glycine and N-carbamyl aspartate is highlighted here because, as I mentioned many times, they are the key intermediates in ribonucleotide biosynthesis. And please note that this fourth subsystem doesn't require the use of palladium, but it's working at the same temperatures and same pH used for other subsystems. Now, if you were really paying attention though, you might have noticed that the output of each system was the input of another. Since they all work under the same conditions, it begs the question whether the entire system would be put together and work in an autonomous way in one pot without any human intervention other than the initial mixing of the components. Let's have a look. If we mix conditions used in other subsystems, so mixing these two keto acids, oxaloacetate and glyoxylate, in the presence of trace amount of palladium and in the presence of prebiotically realistic amounts of hydrogen, bicarbonate, ammonia, and carbamoyl phosphate, what we get? We obtain this messy NMR spectra describing the mixture. Now you can wonder, well, do we get what we were looking for? And the answer is yes. Of course, I probably wouldn't be telling you about this if it didn't work, but indeed it does. Here's an NMR trace of the reaction with all the compounds labeled. It looks very complicated, so I'm gonna show you this overall scheme of what you've seen in the last slide. Here you can appreciate the bigger picture of having a reaction network with 4 different subsystems all working together spontaneously and in autonomous manner. We have CO2 reduction to form it, so carbon fixation, keto acid synthesis of Krebs cycle through aldol condensation and hydrogenation. So we have all the important keto acids. Amino acid synthesis via reductive amination, and in the presence of N-carbamoyl phosphate, the mixture of both N-carbamoyl aspartate and N-carbamoyl glycine, whose carbon backbones become then incorporated into nucleobases. As you can see, this reaction network is large, which can potentially give an explosion of products. And along with these compounds, we also see some other products that could not be identified by NMR. But the products drawn here account for 26% of all the oxaloacetate added as input at the beginning of the reaction. So what's special about this system is, as far as biology like non-enzymatic reaction networks go, we are not aware of any of them that can generate multiple biology-like subsystems without intervention from the experimenter. Most reported synthetic reaction networks need many sequential step changes in reaction conditions, and they resemble less to biology-like system. So in that way, the reaction network shown here is quite unique. It's metabolism-like in terms of its inputs, as it uses hydrogen, CO2, ammonia, and metabolites. It's autonomous and reasonably selective for forming biomolecules. So moving forward, it will be interesting to see how adding additional reactions like phosphorylation chemistry might expand the network towards more and more complexity. Here I just want to mention a few words about the nature of the catalyst. To get insight into this aspect, we turned out for help to our collaborators, Dr. Tutsius and Dr. Song. They did the surface analysis of our commercial palladium that we used in our reactions and confirmed previous studies showing that commercial palladium on carbon is entirely palladium-2 rather than metallic palladium. Taking these results into account, they synthesized palladium-0 and palladium-2, which then we tested in our reactions along with some other commercial palladium samples on different supports. We observed some differences in the reactivity between the various catalysts, but the reaction occurred in all cases. So this demonstrates that the initial oxidation state of the metal and the nature of the support are not critical to catalysis. Before I end, I just want to mention that the use of precious metals in prebiotic chemistry may not end here. The true biological reverse Krebs cycle composed of keto acids was uncovered in our previous work by my former colleagues Kamila and Srijit. They have found that many different reducing agents were able to do the chemistry. But as you can see, these conditions are too harsh to be relevant to the origin of complex reaction networks. So we looked for alternatives, such as precious metals. And here what my colleague Sophia found. She found that rare metals are able to promote bottom sequence under hydrogen using mild temperatures and over broad range of pH. So these results clearly pointing out that rare metal chemistry is not limited but could be applied to broader extent. And this brings me to the end of my talk. In summary, I've described a hydrogen-driven self-organized reaction network that emulates the function of several interconnected biological pathways without intervention from the experimenter, strengthening the link between self-organized reaction networks and the emergence of genetic molecules. I have sketched out a rough path for how rare catalytic elements like palladium could have had many opportunities to interact with carbon dioxide, hydrogen, ammonia, and with some other metabolites, and this way could have contributed to initiate and templating self-organized prebiotic chemistry on the early Earth. I leave you with this open question that we can discuss later. I hope I've convinced you that rare metal chemistry is something important we should consider in prebiotic context as well. With this, I end my talk. I would like to acknowledge Professor Moran, who supervised my work. I'm truly grateful to him and to our collaborators, Professor Martin, Dr. Tutsius, and Dr. Song for their valuable support. A big thank to all the members of my group, especially Kamila and Emily, and to all the people who've contributed in this work. So thank you, Jing and Manialo. Finally, thanks to PCE3 team for inviting us to this platform to give us an opportunity to share our research views and work. And thank you to the audience. It's a great honor to have you all attentive and tuned in. Later, I will be happy to take questions. Thank you once again.
Great. Thank you, Harpreet. That was a great talk. And so, as she said, we'll take questions for her at the end. So next, we're going to be hearing from Ziwei Liu, who is currently an investigator scientist in the MRC Laboratory of Molecular Biology, working with Professor John Sutherland. And he is working on various projects, such as the fixation of CO2 under prebiotically plausible conditions, the synthesis of building blocks, the chemical network for activation of amino acids and nucleotides, and the origin of translation. And so thank you for joining us, Ziwei, and go ahead.
Okay, thank you. So can you hear me?
Yeah, we can hear you.
Yeah,
and we
okay,
can see your
good,
slides.
great. Yeah, thank you. Thank you for the kind introduction and also the invitation me to the PCE3 by Zoom. And so today I will talk a bit about the prebiotic photoredox synthesis for the central carbon metabolism compounds. So before my talk started, I want to leave this question to the audience. So is anybody able to provide me with my favorite food? I think the answer is no, because no one really know about me. So, but in some case, if you know the answer, you know how to provide food for me. But if not, either you can ask me, but I will not answer it yet. So the simple way to provide me the food is you provide everything you can and let me to choose. So in this situation, what I'm going to do is first choose my favorite one. And second, I will choose some food, which is called intermediate. Then I can prepare by myself. The last thing is I can prepare the food from the raw materials. So this is just a story, and I will come back to this story later. So as Kamila and Rita have talking about the prebiotic chemistry, so I have another illustrator for the view of prebiotic chemistry. So I have this point. Yeah, so if we think about from the very beginning, even from the Big Bang, there's only several elements can be formed from the Big Bang. And after that, some small molecules like CO2, which is the character today, and cyanide and other species can be formed. As the reaction network can start it, these things will form the further steps. For example, the amino acid nucleotide, which is the building blocks for the modern biology. Afterwards, the polymerization can happen to give you peptide, oligopeptide, or oligonucleotides, phospholipid, which can be further included in the formation of protocell, then to the LUCA, which is the last universal common ancestor. So this is a broad view for the prebiotic chemistry. As I start my PhD nearly 14 years ago, the first reaction I have done in the lab is using CO2 as a carbon source. And under different conditions, one of the conditions is showing on the screen is the hydrothermal conditions, using iron and just heat at 200 degrees for 3 days. We can get fomate and acetate. So the condition is quite harsh compared to what people said recently, and the yield is just quite rubbish, like 2% or 1%. And if you think about the order of life event, the first reaction only give you 1% yield, and the second reaction starting from the intermediate, like acetate, to something else, give you another 1%. After several rounds, the yield is negligible. So with the reaction, I'm quite happy it works. It's good for me, but the yield is I can't accomplished. So as time passed by and we have learned something from geochemist or geologist, there's a combination from different starting material we need to think about. So the plausibility of CO2, there's no doubt about it. SO2, which is coming from the volcanic eruption, so it's still available on the early Earth and even right now. And the UV radiation, the people like UV radiation, of course, no doubt about that. The people hate the UV radiation, think it can destroy everything you formed. So in both cases, the UV radiation is available on the early Earth. And 2 years or 3 years ago, David Catherine's group have assuming this alkaline lake is plausible on the early Earth and on other planets. So the combination of these 4 stuffs looks quite plausible to us. And when we write the manuscript for this paper, we write something quite carefully about the plausibility, because any people can argue with you about the plausibility. So we said, this suggests that such lakes could have concentrated other probiotic important species on early Earth and maybe Mars. And our co-author Woody Fisher just comment with us, it is really going to be impossible for any body of water at the planetary surface not to fill simultaneously with CO2 and SO2. So the geologist is quite happy about this combination. So we are the chemist. So we just do the chemistry with this mixture. We take the C13 label, the carbon dioxide and the sulfide, adjust to pH roughly 9 and shine in the UV light. What happened? This is the NMR spectra after, I think, 2 hours or 4 hours radiation. So we get something which we can identify as a fomate, methanol, and acetate. And there are some other species. At that time, we don't know what happens. So to make the life easier, we starting from the natural CO2, it's the C13 one, so C12 one. So we don't have these split peaks, and we can identify all the species by NMR by spike with the authentic standards. So they are fomate. This hydroxymethane sulfonate is the sulfonate adduct of formaldehyde, and the methanol, glycolate, acetate, tartranate, malonate, tartrate. And also there's a single peak which puzzled me for a long time. And one day when I discussed with my colleague, it should be the hydrogen. And then we proved it is the hydrogen. The hydrogen yield is really a lot. And even you take the NMR of the UV radiation, shake it a bit, so you can see the bubble coming from your solution. And then we have the carbon NMR also shows there are some other species. It is the oxalate. So oxalate can be found in the Mars right now, but people try to explain why the oxalate is there, and the answer is not that clear, except this reaction. So as I mentioned at the beginning, the yield is quite important because this is just the start point for all the proto-metabolism or other reactions happen. So we should get the yield as better as possible. I try to verify the different concentration of bicarbonate and sulfide, even start from 5 millimolar, which is 100% acceptable by geologists. So the yield is roughly in total 10%. Because if you have this C2 compound, you need a bimolecular reaction to form this CC bond. So this is why the yield for them is really low. I will come back to the mechanism later. Then I increased the concentration of carbonate and sulfide. So we have all these species. The yield is increasing, but still not very good. And sorry, if I keep irradiation for just 1 day, the total yield of carbon dioxide conversion is roughly 70%. And also, there's another scenario for the on the early Earth. So if your carbon dioxide amount is fixed, and when the volcanic just keep erupting the SO2, and your alkaline lake absorbed SO2, so we mimic that condition. We keep the CO2 concentration as fixed and keep flow in the bisulfite solution. So we can increase the yield for the C2, C3 compound to 20-some%, which is quite nice. And people always think the UV, the early sign is not a UV lamp. You can't only use a single wavelength for the reaction. So then we test a broadband lamp source. We call it star lamp, which can provide us the UV wavelength from 200 to 2000. But we use the water to filter out the IR part. So we still get the similar things with I would say a roughly good yield. As the reaction keep going on the early Earth, so the irradiation is keep going on, and your compound is still here. So what happens to each individual product from this reaction network? Then I just introduced each individual species to the system and keep irradiation to see what happens. The interesting thing is, apart from I have present all these compounds, we have found something else. It's citrate, malate, succinate, and hydroxycitrate. So the scenario is, if you have CO2 and SO2 in alkaline lake and keep irradiation, it's not only give you this C2 to C4 compound, but also the central metabolism compounds, for example, the citrate and these things. So as a chemist, we're always interested about the mechanism. So this is the mechanism. Sulfide under irradiation can give you a hydrate electron and a sulfide radical. The electron, of course, is a reducing power, which can reduce the CO2 to the CO2 dot is a radical. And if it go twice of the reduction, it give you formate, then sorry, it's the formate and then formaldehyde and then methanol. And carbonate radical, CO2 radical, can dimerize, giving you the oxalate. The oxalate is a carboxylate which can be further reduced to glycolate and acetate. And if it's still a one electron reduction, giving you this radical. So this radical can recombine with another radical, giving you the C3 compound, which be further reduced. In this reaction network, another interesting thing is the sulfide radical can oxidize the fomate back to this carboxyl radical and keep doing the reaction network, which means the fomate is not a waste product. It can be regenerated to the the bicarbonate radical, which have never been discussed before. And for the glycolate under the reduction can give you acetate. It's different from the reduction I have talking before. The carboxylate reduction is this hydroxy group can be reduced. The acetate is also an interesting point, which can be oxidized back to this radical and get dimerization to the succinate. And this thing, of course, can be oxidized to this radical and get different combination, give you the tartrate malate. And furthermore, if you have the malate radical here combined with another hydroxy glycolate radical, giving you the hydroxy citrate, and after the reduction give you the citrate, and also the citrate can form from another radical recombination reaction. So just simply shining the UV light to the CO2 and the SO2 mixture, giving you all these carbon metabolism compounds. We call it carboxysulfidic chemistry. So the name is coming from carbon dioxide and sulfide. And also, I will come back to the question I have just discussed at the very beginning. So this reaction network, I didn't draw the circle for citric acid because it's only from one compound to some other compound, which just sit there waiting for the reaction. The citrate and this metabolism network is like the intermediate, which can form for example, the amino acid or other species as have just discussed. And the CO2 is like the raw materials, which be provided and used much later. So in John's lab, there's another chemistry called cyanosulfidic chemistry. It's described, you can get 8 amino acid, decarboxy, all normal ribonucleotide, and also the lipids in the under UV irradiation condition. So what is the relationship between these two chemistry, or there's any connection between them? The answer is yes. If the cyanosulfidic chemistry go to the end, it give you this amino acid nucleotide, but if it's disrupted in the middle, it have some precursor. For example, one of the precursor and the majority precursor is glycolon nitrile, which can hydrolyze, give you this glycolate. And furthermore, it can give you the carbon metabolism compounds. And with all these things about metabolism and genetic or metabolism first thing, so what we propose a scenario is like this. So the carbon dioxide reduction and cyanide reduction, or the cyanosulfidic chemistry, carboxysulfidic chemistry can happen at the same time. But because the carbon dioxide reduction only provide this compound which didn't have further more reaction in my reactions, and it just sit there waiting for the further reaction. And the RNA amino acid, which have this capability to do the self-replication and genotype phenotype reactions. So when these amino acid nucleotides try to replicate itself, sometimes the monomer of the nucleotide and amino acid will consume. So how does the RNA world keep doing? They need to find something to provide the monomer and keep to polymerize themselves. Then when they meet the central carbon metabolism compound already present there, they can use this compound to synthesize the monomer of themselves, then use the monomer to replicate themselves, develop the metabolism network. And if this intermediate was also consumed, the evolution pressure will push these proto-cell stuffs to find other carbon salts to be fixed for themselves, to make this metabolism network keep doing. Then they will think about CO2. So this is similar to what I have discussed the story at the beginning. And at the end, I will thank John Sutton to support me to do these crazy reactions. And also thank you for the collaborators and thank you for your attention.
Great. Thank you, Ziwei. And now that all of our speakers have presented, we're going to be opening it up for questions. And so if you have questions, you can either go ahead and type it into the chat, or there's also the feature in Zoom where you can click on the reactions and raise your hand. And if you want to if you want to ask your question directly, you can raise your hand and we can go ahead and unmute you. And so that's another option. So I guess in the chat, I guess Nathan Herwig, I could ask you to unmute if you have your question. Let's see. Give me a second. Yeah, so Nathan, I think you should be able to unmute.
Hello, can you hear me?
Yes.
First of all, I would like to say this is really cool that you know, I'm getting to talk to all these people from NASA, you know. But I had a question about it's like an astrobiology kind of question. I was wondering if gas-based life forms could exist, kind of like how deep sea siphonophores, it's a big colony of organisms. I was wondering if gas-based life forms could exist where it's a big colony of organisms that, I don't know, like can float on the air and yeah, like that. I don't know.
I'm not, do any of our speakers have any thoughts on that question?
I can say something, but priority is obviously to the speakers of today's session.
Okay, maybe I can have a start. So practically, it's difficult to do the gas phase reaction, at least for me. So I didn't really think about that, but it's quite possible to do or to form the gas life or something similar. Yeah, but I don't know if any group in the world have tried to do the reaction. I have no idea about that.
Okay.
Okay, maybe I think I see that there's another question from Kevin Devine. I'll ask you to unmute.
Okay, yeah. Okay, right. Okay, thank you very much indeed, Harpreet and Ziwei, for two excellent presentations with a lot of organic chemistry, which I like as an organic chemist. I got some questions for Harpreet, and actually I noticed Donald Burke sort of read my mind. First question concerns the type of minerals that palladium forms. I think maybe it would be more accurate to use those, in particular, those thought to be present on the early Earth, as well as just pure sort of like palladium catalyst, which we tend to use in organic chemistry to great effect. My other question is about the synthesis of the amino acids. I noticed it was very good. I liked it. And they were certainly present in greater concentrations than the corresponding alpha hydroxy acids, which has been seen before. I have a question about the enantiomeric selectivity of your reactions. Did you get a prevalence of L over D? Was it a racemic mixture for each amino acid? And my final point is about phosphates. If you're going to bring phosphates into these reaction schemes, you should use cyclic trimetaphosphate, which is certainly believed to have been present on the early Earth. Okay.
Okay, thank you for the question. And to answer the first question, yes, we used pure palladium like in our reactions. And coming to the question that can you please repeat again the first question?
Yeah, the type of palladium, you're not going to find it as a sort of pure metal on the Earth. You're going to find it as a mineral, putting an oxide. Would that not be better? Would that not be as well? Although you could, in theory, a complex redox thing involving palladium and more abundant metals such as iron, cobalt, nickel might be more interesting. Because especially if the palladium only works in its low oxidation states like 0, +1, +2, So.
Yeah, okay. So yes, we don't actually need to use pure form of palladium. It's even better if it's combined with other metals like copper, nickel, iron, etc. Okay. So it can induce even like more complex chemistries.
Yeah.
And regarding the oxidation state, in our experiments, we have found that we used either palladium 2 also and palladium 0, and both induce their activity because we have hydrogen around. So it's possible that the palladium 2 is reduced to palladium 0.
Right. Mm-hmm. Okay. And your enantiomeric sort of excesses, if any, or were they racemic mixtures for the amino acids produced?
Actually, we didn't look at this aspect. We analyzed our reactions by NMR, and we didn't dig into this racemic enantioselectivity question. But yes, of course, that's the next thing to evaluate. Right,
because I'm very intrigued by that. I'd love to know, because it may well be that the surface of the metal plus catalyst favors the formation of one enantiomer only, which explains why life on this planet predominantly uses the L form of the amino acid D.
Definitely. And when we have like amino acids around, we can see like chemistries which can go towards biology-like systems. So yeah. This is the next thing that we should look at.
Okay. And finally, I suggested cyclic trimetaphosphates. Have you started using that, bringing that into your reaction schemes to look for the formation of presumably carbohydrate phosphates, in particular ribose phosphates?
Actually, this is our now next step, but we didn't include in this this work now.
Okay, right. Thank you very much. Excellent talks. Thank you. Right.
Great. So I think that probably covered Donald Burke's question, but if it didn't, feel free to retype it into the chat. And so next, there's a question in the chat from Samuel Finney, and he asks, so you've shown us a lot of reactions, but the conditions at which the reaction actually breaks the barrier, I mean the activation energies, and thermodynamic constraints are not discussed. Any thoughts about that?
Sorry, this question is about for me.
I'm not, I guess it might be applicable to both of you. I'm not sure. Do either of you have any input on the, I guess, the activation energies in your experiments?
I would like to read the question again because
Yeah, so maybe if Sam wanted that answer, maybe he could clarify it again or clarify it a little bit. So maybe we could move on to this next question. I think Alex Dean's he wrote one, and also they wrote one. So maybe I'll just ask to unmute if you wanted to do that. You should be able to unmute.
Hello.
Hey.
Can you hear me?
We can hear you. Yeah,
all right.
We can hear you.
Thanks. Sorry. I'm just curious if you're also looking at ways that could lead to spontaneous nitrogen fixation to provide the ammonia to feed in to the reaction networks that you've described in your research, or if it's like assumed that whatever is producing H2 in enough quantities to be driving these reactions would also produce ammonia as a byproduct in sufficient quantities as well.
Actually, we didn't look at the production of ammonia. So, but like, as we recently started to look at rare metal catalysis, so all these aspects we need to look further in our work. So yeah, this is also the next interesting stuff to do, that we have to evaluate ammonia production, but we didn't look at it.
Okay, thank you. Sorry.
All right, thanks. So now we have another question in the chat from Jiaqi Pei. And first, he had a question for Ziwei, and it's how are you able to experimentally prove your mechanisms involving radicals?
Yeah, that is a proposed mechanism. But if you think about in the reaction, we have the C2C3 compound form, and it's a CC bond formation, and also it's definitely coming from the CO2. So what kind of mechanism you can provide or think about to forming this C2, C3, C4 compound? So this is a proposed mechanism, but to prove that, I think it's possible to do the experiment. For example, if you use the it's called ESP, which can identify the radical compound when you're shining the light for the ESP tubes in situ. So, and for our reaction, we just want to report all these possibility and also the yield and all the steps. So we didn't really look to prove the mechanism, but if any group or any other people want to try to prove it, it's fun. And I think it will be a great separate experiment to prove the mechanism is correct. Thank you.
Great. I think Jiaqi also had a question for Harpreet, and it was, what would have been the ambient temperatures around rare metal-enriched minerals on the early Earth, and were they at the surface?
Okay, thank you for the question. So we don't know what were the ambient temperatures around rare metal-enriched minerals. In our work that we used like room temperature 22 degrees Celsius, and our work is inspired from biology. So in biology, we don't see very high temperatures. So on the prebiotic Earth, it's possible that we had the fluctuations of temperatures, so very low temperatures, ambient temperatures even high. So, but in our work, we mimic the more surface chemistry, so more like kind of warm waters or the temperatures that we have today that we consider room temperature. And like we cannot really say what were exactly the conditions on early Earth. But we see biology as if we can learn something from it. So we don't think that if the chemistry that led to life can be very different from the biology that we know today. So if it was a yeah.
Sorry, if I cut you off, you could see what you're saying.
Yeah, I just wanted to say if there is a continuous progression from proto-metabolism to metabolism, so there should be something similar. At least once, until we don't have had those complex organic ligands or complex organics which can stabilize further very sensitive molecules. So on the early Earth, the basic core metabolic networks, maybe they didn't. They should involve also the very, for example, such as I said, oxaloacetate is very sensitive, so we cannot go to very high temperatures.
Okay, great. And so I think this is more of a comment, but from Helen Hansma, she said, wonderful talks. And she wants to point out that indeed trace elements and minerals are worth considering because life started as a trace occurrence and the trace inorganics aren't evenly distributed over Earth or Mars. And I think Kevin also pointed out something to help answer Nathan's earlier question that microbial life can exist in the atmosphere in aerosols and water droplets. And this, I think there's been some proposals that this could be present on Venus at altitudes over 60 kilometers. So thanks for pointing that out, Kevin. And let me see. In the chat, let's see, I think, I guess Nathan Herwig said that he might have another question. So let me see. You might be able to unmute if you wanted to ask a question, Nathan.
So I know that silicon-based life forms are possible, like how carbon is the building block of life forms on Earth, but like, what would that look like? You know, like what would a silicon-based life form like look like?
That seems like a great question. I don't know if any of our speakers have any input on that, any thoughts, or any of the audience?
Yeah, maybe I can try to answer a bit, not straight away, but I got this kind of question from different talks from different people. Just ask how the silicon-based life looks like or how it is formed. So it is possible because the carbon silicon in the same group, right? And the problem for silicon-based life on Earth is not that plausible because the silicon-silicon bond is not as stable as the carbon-carbon bond. But the universe is so big, and maybe some other planet have their condition which suitable for the silicon-based life. It's also possible, but at least for me, I don't know if other people also like this. So at least for me, I'm more interested in the life which already present in the Earth. So at least my research will focus on the carbon-based life. Yeah, thank you.
Great, thanks for that answer. And I think I saw that there's a couple hands raised. I think Helen Hansma has a raised hand, so I'll go ahead and ask you to unmute. Helen?
Kevin Devine can probably answer it more completely than I, but I'm always fond of an article by Stephen Benner in Current Opinion COCB, so it's probably Current Opinion in Chemical Biology quite a few years ago. And he explains very nicely the big advantages of carbon over silicon. So that's all I have to say. Thank you.
Thanks. I guess Kevin also had his hand raised, so I can ask him to unmute as well.
Okay, well, I mean, there's multi sort of factors. First of all, the weak silicon-silicon bond compared to the strong carbon-carbon bond, as Ziwei said. Secondly, silicon has a profound propensity for oxygen. It forms extremely strong bonds to oxygen. Thirdly, that's what most of the surface of the Earth is made up of, silicates. It's a giant lattice where each silicon atom forms a covalent bond to 4 oxygens. So it's not a small molecule like CO2. And the reason for that is silicon doesn't form double bonds, either to itself or to other elements like oxygen. That's a profound disadvantage of silicon being able to form a complex sort of series of chemical structures like carbon does. So the nearest we get to silicon life forms is the medium we're talking through, which is computers and silicon chips. This is a problem. Now, if you had a planet that was unusually rich in fluorine, you may get something interesting happening, but that's for a whole other day. But I hope I've been able to point out 3 reasons: the weak silicon-silicon bond, and in particular, the inability to form multiple double and triple bonds. That's what rules silicon out as an alternative to carbon in complex molecular systems, in particular life. Okay.
Great. Thank you. Was someone else wanted to say something?
What I was talking about earlier about like the gas-based life form was kind of about silicon. Like on a planet that had extremely low gravity, could silicon bond with an oxygen atom and then some other atoms and create small life forms that could form a collective like siphonophores,
I think the rules of chemistry are the same, pretty much, are universal. We see the same kind of silicates on Mars that we do on Earth. As I say, silicon, it forms the bowls. Basically, silicon forms the bowls and maybe even the sort of part of the catalysts that allow complex carbon chemistry to develop into life. As I said, everywhere you go, except at unusually low temperatures, there's evidence of some silicon-silicon multiple bonds, that forming pi bonds, but they're only under extremely sort of low temperatures, conditions which don't occur on planet Earth. And I doubt really that we'll find anything life based on silicon. Delighted if I was wrong, but I somehow don't think we will be, because, as I say, the chemistry of silicon is severely limited because it's a larger atom, and as a result, it can be attacked by nucleophiles. So, for example, silanes, which are combinations of silicon and hydrogen, they're like the silicon version of hydrocarbons. They are readily attacked by water molecules, and they hydrolyze quite rapidly. Unlike hydrocarbons, the smaller size of the carbon atom restricts the ability of attacking nucleophilic species to get in there. So I think it's just constant the size of the silicon atom is basically what goes against it and prevents it from being part of our biosphere.
Great. Yeah, I think there was quite an interesting discussion there. And I think kind of going through the chat, it seems like we might have gone through all of the questions. And so I did see that I think Kamila put some comments on some, I guess, providing some references on ammonia. So she said that if you wanted to reach out to her, you can send her a message after. And I also posted this Slack channel. So I'll put and this is just a channel that anyone can join to kind of continue any conversation. So I'll post the copy of the chat in there so anyone can try and look at any of the comments that people posted. And if we don't have any more questions, I think we could go ahead and thank all of our speakers again for the great talks. So thank you to Kamila, Harpreet, and Ziwei again for the great talks. And thank you all for joining us. And we'll hope to see you at some of the other seminars in the future. So thanks again.
Thank you.
Thank you, James, for this invitation and for organizing and for being our chair today. Thank you to the audience. It's great to have you around.
Great. All right. Bye.
Bye.